plenary lecture. It's my great pleasure to introduce Professor Pascal McKeown, Head of the School and Dean of Education, School of Medicine, Dentistry and Biomedical Sciences, Queen's University, Belfast, UK. Pascal is one of my friends, and we worked together in UK as registrars when I was in UK. He visited Sri Lanka for several times and gave lectures to Sri Lanka Heart Association sessions. So he is going to speak about overview of genetics of inherited cardiac conditions. Professor Pascal.
Well, thank you very much indeed for the invitation to present today. As Dr. Shantaraj has mentioned, we've had a very long number of years of connecting cardiology in Sri Lanka with cardiology in Belfast, and I'm delighted to be able to join you today. I'm just going to share my screen, and perhaps you will confirm that you can actually see. I've got a note here saying that the host has disabled participant screen sharing. Is it possible to release so that I can share my screen?
Yeah.
That's great. Hopefully you can see my screen now. I'd be grateful if you could confirm that you can see my screen.
Yes, we can see your screen.
Okay, super. Thank you very much indeed. So I'd like to talk today a little bit about the work that I do in Belfast, working in inherited cardiac conditions. I'll talk a little bit about chromosomal abnormalities and complex diseases, but most of the talk will feature a discussion around the cardiomyopathies, channelopathies, and aortopathies, which are considered to be monogenic or oligogenic conditions. I'll touch briefly just on chromosomal abnormalities and microdeletions. So if we think about individuals who have extra chromosomes, for example, trisomy 21 Down syndrome, we are familiar that those individuals may have a range of complex congenital heart diseases. Similarly, those who have lost a chromosome, for example, Turner syndrome, have a range of congenital abnormalities, particularly bicuspid aortic valve, aortic coarctation, but also as they grow older, they may develop aortic dilatation and have an increased risk of dissection. We also have other conditions such as Williams syndrome, which is a microdeletion, which features cardiovascular features such as supravalvular aortic stenosis and peripheral pulmonary stenosis, also hypercalcemia and cognitive impairment. And the slide at the bottom right-hand side here shows what's called a FISH assay. On the bottom right-hand side, we can see chromosome 7 with elastin gene and control probes. But in the patient who's affected, we can see that the elastin gene probe is not actually featuring on one of the chromosome 7s, in keeping with a small deletion, which is not obviously available to see unless you use special techniques. I'd like to concentrate on two of the cardiomyopathies, mainly hypertrophic and arrhythmogenic, as examples of monogenic diseases. Hypertrophic cardiomyopathy is characterized by this unexplained hypertrophy. So we can see a post-mortem example here with very marked thickening of the interventricular septum. The pathological hallmark is of myocardial disarray. It's quite a common condition affecting about 1 in 500 of the population, and it's usually inherited in an autosomal dominant fashion. Genetic investigations have really shown a range of genes, mainly beta-myosin heavy chain, cardiac myosin binding protein C, some of the troponins, as well as tropomyosin and actin. So essentially it is now considered to be a disease of the sarcomere. And just a little picture here of the sarcomere showing the thick and thin filaments where most of these proteins are expressed. However, it is a diagnosis of exclusion. So we look for other causes of hypertrophy, be it hypertension, aortic stenosis, athletic adaptation. There are a range of other genetic conditions, including Friedreich's ataxia, other mitochondrial diseases, Noonan syndrome, amyloidosis, and also Fabry disease. And I'd like to highlight just some of the features of Fabry disease and the relevance of excluding that as a condition. Firstly, it's not autosomal dominant. It's an X-linked disease. In the full-blown phenotype, we also get angiokeratoma, as illustrated here. And also one can see changes in the cornea of the eye known as cornea verticillata. There are, however, some individuals who don't really have many of these skin or eye manifestations, what we may call a cardiac predominant subtype. And the reason why it's very important to make this diagnosis is that there are specific therapies now available, including both enzyme replacement therapy and also the use of oral chaperone molecules. Arrhythmogenic cardiomyopathy has traditionally also been called arrhythmogenic right ventricular cardiomyopathy. We do recognize the particular areas of the right ventricle which are likely or more prone to be affected, but increasingly we also recognize that the left ventricle can be affected, and we are increasingly using the term arrhythmogenic cardiomyopathy to reflect that it can affect both right and left ventricles. We may see aneurysms, for example, in this right ventricular angiogram. The pathological changes include fibrofatty replacement. So in panel D here, we can see the muscle in the kind of purple color. The blue indicates the fibrosis and the white, the fatty replacement. Individuals are at risk of developing ventricular arrhythmias. They may have epsilon waves, a late potential. Traditionally, most families have an autosomal dominant pattern of inheritance, but there are also autosomal recessive varieties. So, for example, the unraveling of the genetics of arrhythmogenic cardiomyopathy really centered on this relatively rare disease known as Naxos disease, which is an autosomal recessive disease most commonly seen on the island of Naxos, which is one of the Greek islands. The children have a particular type of hair, but also when they start crawling and walking, they develop very marked thickening of the palms of the hand and soles of the feet, known as palmoplantar keratosis. And that really provided a focus then on the desmosome. So if we look at panel B on the lower right-hand side, we can see the main connections are the adherens junction, the desmosome, and the gap junction. And that's really how the cardiomyocytes interact with each other. The genes that are affected in arrhythmogenic cardiomyopathy are centered really on the desmosome, which is really the part of the cell which actually holds the cells together as they contract and relax. The genes include desmoplakin, desmoglein, desmocolin, and also plakophilin and plakoglobin. And it seems that the individuals develop this weakness within the desmosome such that the cardiomyocytes separate, and that then triggers an apoptotic mechanism leading to replacement fibrosis. It's also clear that individuals with these genetic variants who participate in kind of extreme physical activities are more likely to develop a more dramatic phenotype at an earlier age. And we also have animal models to show that is a working hypothesis. Moving on then to the channelopathies, I'd like to touch briefly on long QT syndrome, Brugada syndrome, and catecholaminergic polymorphic ventricular tachycardia known as CPVT. So long QT syndrome, Brugada syndrome, and CPVT are called channelopathies because the pathogenic variants are generally in the genes which encode the ion channels. So if we look at panel B here on the right-hand side, we can see the action potential. And in the lower part of the slide, we can see the various channels that open and close to facilitate depolarization and repolarization, namely the cardiac sodium channel, calcium channels, and also a range of potassium channels. And these are really important in terms of looking for genetic variants which cause these conditions. So, for example, here is a family that I look after in Northern Ireland with long QT syndrome. The individual who first came to my attention was this girl here, marked as 4-3 on the pedigree, and she presented to the emergency department with a pre-syncopal episode. When we explored the family history, of striking importance was the fact that her aunt, marked here as 312, had died while swimming, and her sister, marked here as 4-4, had also had a near-drowning episode as a young child. The ECG for patient 4-3 is shown here, and you can see that there's quite marked QT prolongation and also QTC prolongation. The QTC, as calculated using the Bazette formula, is here at 5:30. And the shape of the ST segment is also quite abnormal, with this kind of long segment going up to the peak of the T wave. Individuals with long QT syndrome are at risk of developing torsade de pointes, which you can see at the bottom of the slide, and also a risk of sudden death. It's particularly striking, this family history of a death while swimming, and in fact this family has been shown to have a mutation in the KCNQ1 gene known as LQT1 gene. This is a potassium channel gene, and individuals with this particular genetic variant are more likely to have cardiac symptoms during exercise, but also particularly during swimming. The other two common long QT subtypes are long QT2 and long QT3. Long QT2, the genetic variants are found in KCNH2, and individuals with this particular genetic variant are more likely to have symptoms during startle responses, such as alarms going off, the doorbell going, the telephone going, whereas those who have long QT3 have variants in the cardiac sodium channel, where events are more likely to occur at rest. You can see that there are a very large number of genes which have been linked to long QT, but the three main ones are long QT1, 2, and 3. Brugada syndrome is characterized by these changes in the right precordial leads, and we're looking particularly for a type 1 Brugada pattern with the marked ST elevation, the coved ST segment going down to a terminal negative T wave. Lots of different genes have been reported, but there's only convincing evidence really for the cardiac sodium channel SCN5A. Indeed, we have a very large family in Northern Ireland with a particular variant of the sodium channel gene, which allows some individuals to have long QT syndrome and others to have a manifestation of Brugada syndrome. However, in the majority of cases, it's thought that this is more likely to be oligogenic. That is, it requires a number of mutations in different genes to be present rather than being simply monogenic in most cases. The final channelopathy I'm going to talk about is CPVT. Here's a classical exercise test response. Most patients with CPVT will have a normal resting ECG and normal echocardiogram, but when they start to exercise, they develop initially ventricular ectopy, and then the hallmark is in the middle here, which is this bidirectional polymorphic ventricular tachycardia. There are both autosomal dominant and autosomal recessive variants, and these are generally in genes linked to calcium movement within the cell, including the ryanodine receptor and calsequestrin. Now, the aortopathies are also quite complex. A range of syndromes such as Marfan, Loeys-Dietz, and Ehlers-Danlos. But we can also find familial forms of aortic aneurysm, And these are typically linked to a range of genes. In Marfan, for example, it's the fibrillin gene. In Loeys-Dietz, transforming growth factor beta receptors. There are actin genes, Notch genes, a whole range of other genes that are implicated here. And indeed, within the service in Belfast, whilst we do the cardiac screening for these patients, the geneticists actually look after the aortic gene genetic screening or testing. Finally, just in terms of complex diseases such as coronary artery disease, we know that there are various risk factors such as hypertension, smoking, hypercholesterolemia, diabetes. But we also recognize in some families this very early onset, suggesting that there is a clear genetic predisposition. However, the influence of any one single gene is relatively small, perhaps an increase of something like 6 to 17% for each genetic variant. This is known as a Manhattan plot. And in this case, using samples from over 22,000 cases, 64,000 controls, and then validating these signals in 56,000 other individuals, we're able to identify a number of genes which are really above the line here, which suggests that these genes may have some influence, but each individual gene only has a very minor increase in risk. Now, we also are able to offer what we call molecular autopsy. So typically where there's been a sudden death, the pathologist would have looked at both the microscopic and macroscopic features. In the paper by Barry Marin, Sudden Death in 387 Young Athletes, he reported that hypertrophic cardiomyopathy was the most common cause, but you can also see things like Marfan syndrome, ARVC, long QT syndrome. But around the same time, there was also a publication from Australia which suggested that about a third of these cases are unexplained, known as SADS. And it is now possible to offer genetic testing at post-mortem as long as you've got a DNA sample from the person who died. And we are able to identify variants in various channelopathies, for example, long QT, and particularly CPVT, which you wouldn't see either on histological or on macroscopic examination. Genetic technology is becoming in many ways simpler, but also at the same time more complex. We can do next generation sequencing, looking at specific panels of genes rather than just doing an individual gene. And we're also moving to developing sequencing of the exons, but also potentially whole genome sequencing. The challenge, however, for us is actually interpreting these genetic variants. We can classify these variants as 1, 2, 3, 4, and 5, with 5 being benign and 1 being pathogenic. Unfortunately, many of these genetic variants end up in class 3, i.e. a variant of uncertain significance. We can look at population data, looking at the frequency of these variants. We can use computers to try and predict whether or not they will have pathological consequences. We can look at segregation within families. Is it a de novo, i.e. a new mutation within the individual where the parents are not affected? But it can be quite difficult to interpret these genetic variants. There's a very good website which I'd suggest you have a look at, known as Online Mendelian Inheritance in Man, which provides you really with a huge range of different types of information about genetic conditions. And if you've got any concern that the condition that you're looking at might be genetic, you simply type that into the OMIM website and you'll get lots of information. So in terms of the Inherited Cardiac Conditions team, we are a multidisciplinary team, including cardiologists, both adult and pediatric, with a number of specialist nurses. We also have geneticists, the staff from the forensic teams if there's a sudden death, and also genetic counsellors. We take a detailed personal and family history, look at all the past investigations. We provide information. We offer genetic testing where it's appropriate. We then look at phenotype-genotype correlations, looking for segregation. And then if we identify a pathogenic variant, we can undertake cascade screening through the family. So we can identify those branches of the family that are at risk and those branches of the family where no longer-term follow-up is really required. So in summary, inherited cardiac conditions affect probably about 1 in 400 of the population. A multidisciplinary team approach is essential. Genetics offer diagnostic and, on occasions, both prognostic and therapeutic options. Cascade screening can be very cost effective in terms of limiting the number of family members that need to be kept under long-term review. And we have increasing patient expectations around personalised medicine about how the genes will help inform, I say, both the therapeutic and prognostic information we can provide. So thank you very much for giving me the opportunity again to present today. It's been delightful to have this opportunity, and I'll stop sharing my screen now.
Thank you very much, Professor Pascal McKeown, for that very informative lecture in cardiogenetics. I think we have a few minutes to answer any questions. So may I just ask a question and pass a comment as well? So when I make a preliminary contact with Professor McKeown to have this lecture organized, he asked me a question whether there is any cardiologist is dedicated to genetics in Sri Lanka. I said no. So can you just, Professor McKeown, highlight further about the need to have a specialized cardiovascular genetic program? And especially in Sri Lanka we don't have, but your question was an eye-opener to us. Then I put that question across to our council to need to highlight that there's a need. One of us should be interested, take a special interest in genetics. Thank you very much for putting that idea across to us.
Okay, we can help in any way. We're very happy to offer advice and support. In terms of how we run our service. It is becoming increasingly complex, but I must say it's a really fascinating subspecialty within cardiology now, and of increasing interest to our trainees as well.
What's the advice you give to set up a genetic unit in the cardiology setup?
It's very much a multidisciplinary approach. I mentioned you do need both adult and pediatric cardiologists. Specialist nurses are very good in terms of building the family pedigrees and doing a lot of the detective work. But also we need input from our genetics teams. The concern that we have at the moment is that a lot of people will go off and get their genetic tests done, but the interpretation of it is very complex. And I think in the past, we have made mistakes in informing people that they've had a genetic mutation, which subsequently turns out not to be a mutation at all. So we do need to be very cautious here. But I say it is very much about multidisciplinary teamwork and getting all the different experts together. So the technology, I say, produces lots and lots of information, but it's really the bioinformatics support that's required as well in order to interpret the genetic variant. And it takes quite a lot of time to explain to patients and their families some of the complexities around this. So time is also important. But because these conditions do carry a significantly increased risk of dying suddenly, we think it's very important to invest in this type of service.
So setting up a genetic lab, what are the initial things you advise, Pascal?
Yeah, so I mean, it depends. Actually, we don't do many of the tests now in Belfast. We had originally done them in Belfast, but there are expert centres really across the world which can do this with very high throughput and will also do the bioinformatics support. So, for example, most of our work is now sent to supra-regional centres in England. For example, Manchester, most of our samples now go to Manchester. But I am aware, for example, in some other countries, there's a very good genetics service in the north west of Spain with excellent bioinformatics support. And I know that some of my colleagues in Dublin, for example, have been sending their samples to these specialist labs, which don't just do the genetic testing, they also do the interpretation for you. So it should be possible, if you were interested in looking at the opportunities either for testing in country or potentially looking at specialist services outside the country. The one thing that's really important, however, is always to look at the genetic variants based on the correct population. Many of the labs that started this work had data sets only for Caucasians and completely misinterpreted much of the information because they didn't have panels which reflected genetic variation based really on different ethnic backgrounds as well. So it's really important to ensure that you're comparing like with like. And that was one of the early lessons. There's been quite a lot of work coming out of North America, for example, from the Mayo Clinic, I suppose, producing guidance around how you might develop genetic services, which I can link you to if that's of any help.
So, how do you do, list in the priority-wise the cardiac, heritable cardiac conditions which are medically actionable in terms of priority? So, mainly in terms of offering possibility of offering the therapy and maybe prognostic information in the present availability of present day accurate genetic panels.
Yeah,
what
so
are the
for example... Yeah, so some of the channelopathies can be quite difficult to diagnose, particularly CPVT. So we've been able to offer CPVT. We've got several families in Northern Ireland. We have one family where 6 individuals within the family have died before we reached the diagnosis, but it's now possible to offer cascade screening within the family. For a lot of the hypertrophic cardiomyopathy individuals, it may not change their immediate management, but it has very significant impacts for the rest of the family. So what we call cascade screening allows us to identify branches of the family that are at risk. It also allows us then to exclude large numbers of these families to say, well, actually, you're not at risk of carrying this gene, and therefore you don't need longer term follow-up. So I think there are cost savings as well. It allows us to limit some of our screening services then to those who are at genetic risk rather than having everybody within the family being screened. So we use our genetic testing for our cardiomyopathies and channelopathies, and we're very fortunate to have widespread access to very good genetic testing, which we believe to be actually cost effective in the longer term.
Pascal, for that illuminating lecture. And from our point of view, please convey our regards to your team in Belfast. Actually, yesterday, Dr. Ganesh Manoharan joined us for Tava session. I think please take our compliments from Sri Lanka to Belfast.
Okay,
we hope that
Dr. Matharan and Dr. Thakkar, I thank you very much again. I really enjoyed the opportunity and hope to see you again in person at some stage. So thank you very much again.
Thank you, Pascal.
Thank you.